Today we're going to talk about the male reproductive system. We'll start out with the main product of the testis, which is the spermatozoon. We'll look at some components of that. And then we'll talk about the testis, spermatogenesis, the excurrent ducts, the accessory glands, and the outlet of sperm throughout the male reproductive tract. Medical School Histology Basics, the male reproductive system. Hi, I'm Larry Johnson. I'm a professor at Texas A&M University, and today we're going to talk about the male reproductive system. We want to conduct a histologic examination of the testes, which produce spermatozoa, the male gametes, the excurrent ducts, which transport and mature sperm, accessory glands, whose secretions support the viability of sperm, in the male reproductive system. We also want to learn the structures to facilitate the male gonad to produce an endocrine and exocrine secretions, endocrine being testosterone, exocrine being the spermatozoon. We're looking at the spermatozoon, then look about spermatogenesis, how it was produced, support cells in the testis, Sertoli cells and Leydig cells, and talk about hormonal control, epididymal function, and accessory glands is what we will talk about. Now, the function of the male reproductive system is the testes produce both spermatozoa, which is the exocrine secretion, and testosterone and endocrine secretion. The function of the male reproductive system are to produce, maintain, transport spermatozoa, the male gametes, and protective fluid, semen, and discharge the spermatozoa containing semen within the female reproductive tract. So that's what it has to do. It has to produce sperm, has sperm mature, as it go through the epididymis, accessory glands, and ultimately deliver it into the female reproductive system. Here we see the male reproductive system composed of the testis, the epididymis, the ductus deferens. Comes back here, you have seminal vesicles coming into here. There's a prostate, so you got a prostatic gland, and then it goes to the urethra. Here's a vulvourethral gland right in through there that pushes the urine out before ejaculation occurs, and then urethra that goes to the outside. And here the testis, the human testis, you can see where a tunica albuginea or white coat has been reflected back, and you can see the white coat. Inside there's a series of tubes. These are the seminiferous tubules, and there is a capsule that surrounds the testis, as you see here and here, and from that it sends in some septum that subdivides the testis somewhat, not completely. But here you see a seminiferous tubule deflected back, and that runs into the rete testis tubule as part of the mediastinum area, and then it goes to the various efferent ducts, which then goes to the ductus epididymis, and from there on it goes to the ductus deferens, which ultimately goes into to the prostate and then into the penile urethra. So here we see the epididymis, the tail of the epididymis, ductus deferens, and here's the spermatic cord where nerves, blood vessels attach to the body. If we look at the testis, we see there's a thick capsule here, and here's the seminiferous tubules, and between the seminiferous tubules would be the Leydig cells located into here. So if we look at the human testis, we see the tunica albuginea reflected back, and it is dense irregular connective tissue like all other capsules of the body, and you can see the fibroblasts and the collagen in through there. Also, you can see the lining. The outside lining has a mesothelium. We can see it here on this slide, that slide, and it's also right over here. There's also blood vessels that surround the testis inside the capsule, and they go around to kind of cool the blood. See the arteries, veins, see the valve there. There's also some lymphatics that we can see here and here. Now, the main product of spermatogenesis is the spermatozoon, and here we see the spermatozoa, and this happened to be of a horse. Here's another one here, a scanning EM view of it, the middle piece, the principal piece. And there's mitochondria around the middle piece. You can see those here. and you can see it here. Now, if we look at the spermatozoa, we can see the nucleus, which is the main part, and there's a series of membranes. There's a nuclear membrane, the interauxomal membrane, outer auxomal membrane, then the plasma membrane that circles around the entire sperm. And also, if we look at the spermatozoon, which is one, we can see the middle piece have mitochondria, also have dense fibers, and then it has an axoneme. And the axoneme continues all the way through to the end, where here we see some doublets and singlets, and then you only have singles at the very, very end piece. So you have a system by which you have mitochondria to provide energy. You have a fiber sheath which is flexible, and you have an axoneme which facilitates the movement of the tail by the dining arms and the doublet sliding with one another. And the spermatozoa is produced in the testis. Here we see the human testis, the seminiferous tubules therein, and we see the Leydig cells between those. You see the capsule and the septum that's coming up between here. We see the Leydig cells. In the human, the Leydig cells have a rachichryssalid structure. Don't know exactly what it's does, but it's there. And here we see a seminiferous tubule, which has germ cells of different sizes. The spermatocytes here, and the spermatids over there. Now, in the fetal testis, we just see gonocytes, and the gonocytes give rise to this spermatogonia, which ultimately divide and differentiate to produce spermatozoa. Here we see the Leydig cells, and we actually see some lipid in the Leydig cells and the seminiferous tubules. And you can see a host of germ cells, these round ones, these bigger ones, primary spermatocytes, spermatids, and elongated spermatids that you see there. Now, testosterone needs to be higher concentrations in the tubules than the rest of the body, and therefore you have Leydig cells distributed throughout the testis. So in the seminiferous tubule, we see germ cells of different types. You've got a spermatogonia on the base, spermatocytes above there, and then you got spermatids. And here we can see the spermatids in through there, spermatocytes over here. And then also we've got a Sertoli cell. Here's a Sertoli cell nucleus here, kind of pear-shaped. You can see them here and here. These are nuclei of the Sertoli cell. So in the tubule itself, you have germ cells and Sertoli cells, and surrounding around the tubule, you have myoid cells. So we see fibroblasts throughout the interstitium, which is what's between the tubules. You see the Sertoli cell nucleus, and we see the myoid cell nuclei in through here. A myoid cell marks the limit of the outside of the tubule, and then we have spermatogonia. Sometimes they're dark, sometimes they're light. If we look at this light microscopic view, you can see endothelial cells, there's blood vessels. Anytime you got endocrine function, you're going to have a rich vascular supply. Here we have spermatogonium. There's a Sertoli cell nucleus there, and there's one here, a nice pear-shaped Sertoli cell nucleus. And then we see the spermatids. So these are nuclei spermatids, and we see a little cap, acrosomic vesicle, and a little cap phase spermatids. And these are the ones that look like bullets. These are the ones that mature. So you got spermatogonia, spermatocytes, and spermatids. And we can see those in a drawing and in the real tissue. Here we see the spermatogonia, the pale ones, but there's also dark spermatogonia too, again on the base. And then we can see above the base we have spermatocytes, and we can see they vary in type 2. These are younger than these ones. These are pachytene primary spermatocytes. And then there's secondary spermatocytes. You can see the secondary spermatocytes here. And then when they divide, they give you spermatids. And the spermatids come out spherical and look very similar to the secondary, except they're smaller. The chromata looks very similar, but they're smaller, half the size. And then we get mature spermatids. Mature spermatids is a differentiation of the round spermatids, the young spermatids that we see there. There's also Sertoli cell nuclei. There's one there, one there, one there. one here as well. And there's myoid cell nuclei, as we can see, and the myoid cell marks the limit of the tubule. And so if we look along the base where the spermatogonia are, we can see mitotic figures. So these are mitotic figures in the spermatogonia, as opposed to these guys. These are meiotic figures where you have dividing spermatocytes. So dividing spermatogonia versus dividing spermatocytes. And it has to do with whether they're off the base or whether they're up off the base in terms of location in the height of the seminiferous epithelium, and because as they move forward, they mature. So here we can see the myoid cells around the seminiferous tubules. We see Leydig cells, lymphatics, very thin wall. See capillaries, blood capillaries, Leydig cells. You can see some lipid and Leydig cells here, and we can see more Leydig cells, seminiferous tubules, lymphatics inside there. Since developing germinal cells require 100-fold concentration of testosterone than others, that's why you have its close proximity. I think we said that one time before, but we want to confirm that. Lymphatic artery, you got fibroblasts, primary spermatocytes, myoid cells, mitotic figures of spermatogonia giving rise to spermatocytes. If we look at a Leydig cell here, you can see it has a lot of smooth cytoplasm associated with it, and it also has some lipid. But if you look at that at the ultrastructure level, what you see is a smooth endoplasmic reticulum, some lipofuscin, some lipid, but there's a lot of smooth endoplasmic reticulum associated with it producing testosterone. Also, the mitochondria have a tubular type cristae. Instead of having a typical lamellar, it's little tubes, and so the cristae is tubes. Maybe there's a macrophage in the interstitium. There certainly would be macrophages in there. And here we can see a high voltage electron micrograph showing the Sertoli cell nucleus here, and here another Sertoli cell nucleus there and there. And you can see that the Sertoli cell is like kind of like a tree. Sertoli described it first with branches. They're not really branches, they're more like bed sheets that surround the germ cells. So the germ cells are not inside the cell itself. They're in like a little pocket of the cell. So we see the spermatocytes, spermatogonium here, spermatocytes, this happen to be in a horse, and then these are spermatids in here, and this is the elongated spermatids. You can see the manchette. If we look at this later, we can see the spermatid nucleus, the Golgi apparatus, the centriole giving rise to the developing tail. And then finally that tail gets more developed, as you can see there, and there's a manchette that may be associated with elongation of the nucleus and certainly helping development of the tail. So this spermatid is the same as these guys here, with a little acrosomic vesicle forming up there. So these are Golgi phase because of the Golgi phase spermatids. Here we see electron micrograph of the Sertoli cell, which is a tall cell again. It's from the base all the way up to the lumen. We don't see the lumen up there. And we see two primary spermatocytes. This is one, the nucleus over here and the nucleus over there. In this one, we can see the synaptonemal complex, which is where you have crossing over of the DNA during meiosis to occur. But of the Sertoli cell, you see the bizarre shape of the Sertoli cell nucleus, a little bit of nucleus up there as well, euchromatic nucleus. We don't see the nucleolus, but there usually is one. We can see lipofuscin, heterophagic vacuoles that have been eaten up, very long microtubules in through there. Mitochondria are long and look quite a bit different than the mitochondria from the spermatocytes. A nice Golgi apparatus that we see for the Sertoli cell. So the Sertoli cell will be touching each Sertoli cells on the sides where they make a junction. And we can see here where if this is one Sertoli cell in purple and another one over here, then right here there is a barrier between these two, and this is the Sertoli cell occluding junction barrier that's above the spermatogonia. The spermatogonia are in the basal compartment, and then these cells are in the adluminal compartment. Primary spermatocytes and spermatids have never been seen by the immune system, and you need a barrier to keep those new cells which appear after puberty from gaining access to the immune system or vice versa. And here you can see an electron micrograph showing you that barrier where lanthanol nitrate was able to penetrate in underneath there, but then stopped abruptly right here at the occluding junctions of the Sertoli cell, which separates the basal from the luminal compartment. There's no counterpart to this in the female, because when she's born, she already has the primary oocytes. What happens is that the prelapitine primary spermatocytes, which are produced from the type B spermatogonia dividing, they migrate through the barrier, and that's how we get things from the basal to the luminal compartment. Now there's three types of germ cells. You've got the spermatogonia, spermatocytes, and spermatids, and these divide spermatogenesis into three major events: the mitotic activity or spermatocytogenesis, meiosis, and spermiogenesis. Those are the three things. So we have spermatogonia divided by mitosis, spermatocytes divided by meiosis to make different individuals, and then you have differentiation of the round cell to that looks like a sperm. And here we can see the changes that occur, and we have named these different ones. If you look at it in human, you can see the Golgi phase with the archosomic vesicle, the Golgi apparatus here, the cap phase where they has a little cap over it. The archosome has formed a cap. Elongation phase where you get elongation of the nucleus and the manchette present, and then finally the manchette disappears, mitochondria moving around the tail, and you get the maturation phase. So these are the different phases, and you can see how the development occurs as it repeats back in this one stage of the cycle. Spermatozoa are released in stage 2 of the cycle. These guys are released. And so here's a human spermatozoan. You can see the mitochondria in the middle piece. It's having a little bit of cytoplasmic droplet. But here we see the nucleus and the host of membranes with the archosome on this region. Now, so spermatozoa is produced in the tubules. It goes through the rete testis tubule and then goes through the efferent ducts and then to the epididymis. And that's what we're going to see. Here we see the epididymis uniform, but it has stereocilia on its surface. Now, the seminiferous tubules give rise to the rete testis tubules, and this whole region is known as the mediastinum. So here we see the mediastinum, where the spermatozoa are going from the seminiferous tubules out through the rete testis, through the efferent ducts, and here we see the rete testis tubules in through here, as we can see, and here we can see again, we can see seminiferous tubules over here, the mediastinum area where you have rete testis tubules. These have simple cuboidal epithelia on there, which are ciliated. I have one cilium on each one. And then we have the efferent ducts. Efferent ducts is true ciliated cells, and that's a counterpart to the oviduct in the female. And then we have the ductus epididymis. And so here is a single ductus epididymis with a pseudostratified columnar epithelium with stereocilia. And so that's how sperm get out from the seminiferous tubule to the rete testis, efferent ducts, and then to the epididymis. And here we can see the efferent ducts, true ciliated cells on its surface. You can see the cilia here, and the cells have varied in height, and as a consequence, they look scalloped, and so you get a scalloped appearance of the duct. So here's a scalloped appearance of the efferent duct, true ciliated cells, and then that gives rise to the ductus epididymis. Here we see some spermatozoa in there and the long stereocilia, not true cilia, just very long branch microvilli that holds the sperm off of the epididymis epithelium, and you can see there's some smooth muscle here in the head of the epididymis. So here we see the head of the epididymis, pseudostratified columnar epithelium with stereocilia is what we see there. Some more sperm in the lumen. This is actually a blockage in the duct, and they expect to see some macrophages in there too. You see one right here, as well as the sperm in through there. Usually you don't have sperm stored quite so concentrated in there, but there was a blockage in this case. The tail of the epididymis, you have more smooth muscle. As you go down, you can see the smooth muscle here around the epididymis, and usually the height of the epithelium is reduced as you go through the epididymis. And if you look at one of those epididymal cells, you can see the stereocilia and see that indeed it is branched. The branched, very, very long microvilli are known as stereocilia, and you have them in the epididymis. There's a lot of Golgi apparatuses in these epithelium, not sure why, and also a big swirl of smooth ER mitochondria that we see in these cells. Now, in the epididymis, as you see here, there's head epididymis, body epididymis, tail epididymis, and the ductus deferens. Sperm gain their ability to swim, and they also become fertile. You have maturation of the plasma membrane, mitochondria become stabilized with disulfide bonds, and the nucleus DNA changes a little bit. It too is stabilized by disulfide bonds during the transit in the epididymis. And here we can see the ductus deferens, which has longitudinal muscle on the inside, outside, and circular muscle in the middle. So there's lots of muscle in through there. The height of the epithelium is not as high as it was in the epididymis. And then we see this one here, we can see the smooth muscle on the ductus deferens. It's happened to be a spermatic cord, so you see blood vessels and nerves and lymphatics in through there, but this is the ductus deferens that go through there, smooth muscle, enfolding of the lamina propria and the epithelium, as you can see there, still pseudostratified columnar epithelium of lower height. Now, how does sperm go through the tract? In the testis, just bulk flow. Maybe some contractions of the myoid cells, but basically the seminiferous tube produce fluid and just wash the sperm out. Same is true for the rete testis, except the rete testis has one cilium, it's very long. The efferent ducts, there's probably cellular reaction and then bulk flow. And then the epididymis is contraction of the smooth muscle, and certainly the contraction of the smooth muscle in the ductus deferens that facilitate the contraction. And the contraction in the smooth muscle there is much faster than what one would see, say, in the gut. And that's because each of the smooth muscle cells are innervated to coordinate the activity of the ductus deferens whenever ejaculation occurs, because the sperm are in the epididymis until the right end of the process of the starting of the ejaculation of the. And so here we see the testis, epididymis, ductus deferens, and the spermatic cord. Here we see a cross-section of a spermatic cord, ductus deferens there. You see rich vascular supply. This is blood vessels, nerves, lymphatics, with the ductus deferens as well, with smooth muscle, the inner circular, outer, and most inner longitudinal muscle, middle circular, I should say. And so through the ductus deferens, sperm come, and then they go into the ejaculatory ducts of the prostate. Here we see the prostate in through there. Seminal vesicles are also there, dump their contents into here. And then it goes out through the urethra. Vulvar urethral muscle clears the urine out before the ejaculation process. And it's really interesting fluid. It can stretch it. If you ever tried it, you can actually stretch the fluid, which normally you can't stretch water, but you can the fluid. That's a pre-ejaculate fluid. Just a prolonged erection will yield that fluid. And so here we see the ductus deferens, and we see the seminal vesicles. Seminal vesicles have this honeycomb type shape. A structure. There's more glands and less connective tissue in the seminal vesicles than in the prostate. And here we see the honeycomb, and you can see the epithelial cells on the surface. These are columnar epithelial cells, tight junction between adjacent cells, and they secrete their fluid, seminal fluid. And so in the prostate itself, we have the prostate here. You got the urethra through there, and the ejaculatory ducts. Where the sperm would be coming through from the ductus deferens that we can see. Here we can see in the prostate, we can see the secretory cells in through there, and a characteristic of the prostate is that there are smooth muscle cells in the connective tissue. So the more heavy pink that we're seeing is actually smooth muscle cells. Here you can see a corkscrew-shaped nucleus there. And then there's cross sections of smooth muscle cells that we can see, but this is the lumen and the secretory epithelium, which varies in height. Again, we can see the prostate here. I see the connective tissue and muscle here, which is a greater amount than the secretion part in the prostate compared to the seminal vesicles. Again, we can see that here, and then here we see the penis, and this we have the erectile tissue here and here. This is a corpus cavernosum, and there's a band of connective tissue that surrounds it like this, and there's also connective tissue that surrounds this. This is a corpus spongiosum and urethra, and what this does is erectile tissue holds urethra open during erection so that the ejaculation process, a sperm can go through. But this is erectile tissue in through here, and so if we look at a cross section of the human penis, you can see the erectile tissues in through here and here, and you see the band of connective tissue, and this connective tissue is shaped in like a cylinder, and so that the penis projects out as opposed to inflates around like a balloon would. And then here we see the spongy cavernosum, which holds your urethra open during the process of while you have an erection. In the urethra, you have transitional epithelium that we see here and here, and we also note these big nerves that are coming down through the testis, and there's through the penis, and then here we can see a smooth muscle as well as connective tissue around. So in summary, the male reproductive tract it functions to produce and deliver male gametes. That's what it does. It has a testis, the epididymis, ductus deferens, the seminal vesicles, prostate, urethra, in order to get that done. We have some questions on the male reproductive system. Blood-testis barrier is created by sartorial cell to sartorial cell desmosomal connections. No, not desmosomal, tight junctions. Blood-testis barrier has no counterpart in oogenesis. Correct. Prevents the movement of prelapitine primary spermatocytes from the basal to the luminal compartments of seminiferous epithelium. No, it does allow it to do it, even though there's a barrier. It allows it. So the answer is B. This one is true. It doesn't prevent movement, it allows it. So this is the only one that is true. The other ones are false. The wrong junction here, and it provides, not prevents. Which organ mechanism of spermatozoa transport does not match? Seminiferous tubules, bulk flow, yes. Rete testis, bulk flow, limited ciliary action, yes. Efferent ducts, bulk flow, ciliary action, yes. Epididymis stereocilia action, no. Stereocilia does not move, and so that cannot move sperm alone. So the answer is D. Meiosis occurs during spermatogenesis, yes. Oogenesis, yes. Asexual reproduction, no. So the answer is D. We want to thank the various books and sources for original drawings and some pictures that were here. We want to thank them because they made those. I did not. I just use some of those to illustrate some of the things that we have, and we want to thank the original source. This is Stockholm, Sweden. I went there one time. This is the Vasa, where there's a ship that had 2 sets of cannons that sank on its maiden voyage, and they brought it up and put it in that museum there. A 300-year-old wooden ship that survived. And this is where the Nobel Prize is given, Stockholm, Sweden, in City Hall. This is City Hall right here. This was our hotel that we see, City Hall. And they have a center room. Center room, they get the Nobel Prize, and the one to the right is laced in gold, and they go dance in the gold room after they get the Nobel Prize. I've danced in the gold room. I didn't get a Nobel Prize, but I certainly danced in that gold room. So that's the end of the male reproductive system. I hope it was useful to you. If it was, please share it with your colleagues that maybe they can benefit from it as well. Thank you.